Hello. Very nice to see you all here. We are Boston Taiwanese Biotechnology Association, BTBA. I'm Annie Chen, the co-director of academia team working with 𝗦𝗽𝗲𝗍. Welcome to our very last academic workshop this year before our annual symposium, which will be held on July 23rd and 24th in Boston. And this year is going to be very special, not only because it is going to be an in-person event after the 2 years of virtual ones, but also because it's the 10th year anniversary of BTBA. So you can find the registration info on BTBA's official website from May 1st. And we are now taking abstracts, and we will invite 3 people to the meeting to give our presentations. Most importantly, the invited oral presenters will be awarded free tickets of the symposium as well as a gift card. So don't forget to submit your abstract by the deadline. And for our BTBA podcast, we recently invited Dr. Xianrong Zeng and Dr. Darren Du to share their stories. And I have already listened to the 2 episodes, and I would say the stories and the speakers are really inspiring. So go check it out if you haven't. And next Friday, 3:30 p.m., BTBA and Harvard Taiwanese Student Association will be holding a special event, Meet the Experts. In the event, you will learn from 2 experts in the fields of AI and biotech from Portugal about their career journey in academia and entrepreneurship. After all these announcements, it's time to introduce our moderator for today, who will introduce our 2 excellent researchers to us. So Dr. Jason Liu, 𝗧𝗶𝗺𝗲𝗍, received bachelor's degrees in chemical engineering and civil engineering from National Taiwan University, and master and PhD degrees in biological engineering from Cornell University. So currently he works as research fellow at Boston Children's Hospital and MIT. And his research focuses on drug delivery, cell therapy in diabetes treatment, and mRNA therapeutics. So let's welcome Jason.
Thanks, Annie, for the great introductions. Hi, I'm Jason. So before I start the seminars, just the housekeeping rule I want to announce. We will not have any questions during the presentations. All the questions will be asked after the presentations. We have the QA sections. You can either use the chat box to type in your questions or using the link for the QA sheet. Okay, so first, I want to welcome all the people to join our the last BTBA seminar this year, and we have 2 outstanding scholars. One's far away from the UK, and one's from the Johns Hopkins universities. And you guys might feel we why both of us be together in these seminars. And I found one interesting thing, we have something in common. And you might think about hey, you guys are all dudes. Yes. And you guys all from Taiwan, right? Or you guys all study from National Taiwan University. That's true. So, but the most interesting is both of us took the economic course at NTU. And why this is so interesting is because I can remember when I first took the microeconomics at NTUs. That's the first time I heard about the words called the neuroeconomics. The first thing came into my mind will be why neuroscience has any relationship with economics. But after I took the microeconomic course, especially after the game theories, I realized well, we have one organ composed of a lot of neurons. That's our brain. Our brain can help us to think, to have the emotions, and most important is help us to make the decisions. For example, after the seminars, you guys might think about where should we go for dinners, or like what kind of car we want to buy. We can choose from Toyota to Mercedes. That's all decided by our brands. And our brands can help us to make the decisions in a lot of like the economy activities. So if you have any business or you plan to start your own business, maybe Dr. Fei-Yang Huang's talk will be benefit for your business. Okay. So before Dr. Huang start his presentation, I want to first introduce him. Dr. Huang received his the MD degrees and minor in economics from National Taiwan University. And that's the two keywords, MD and economics. And after the MD degrees, and he went to the UK to study his PhD in neurophysiology from University of Cambridge. And currently he's a postdoctoral research associate at University of Cambridge. And it's great to us is we can have an outstanding scholars presenting his work far away from the other Cambridge to the Cambridge here in the United States. And let's welcome Dr. Huang, and now the stage is yours.
Thank you very much for your introduction. Let me first share my screen. Okay, can you see my slide? Okay. Oh, thank you very much for your introduction, and also thank you very much for inviting me over to share a little bit of my research in economic decision making. So my name is 𝗧𝗶𝗺𝗲𝗵. As Jason introduced, I received my medical degree from NTU, and I recently received my PhD from from University of Cambridge. And my research focus on how the brain makes decision making, because decision making is very complicated. So we need to combine experimental and computational approaches from multiple disciplines. And recently, I'm very interested in bringing my research results to clinical applications. So how our understanding of the electrical properties can help treat brain disorders. So that's what I'm going to share with you, and hope that can introduce this exciting field with all of the audience. So first of all, let's see an example of how can we treat the brain disorders by using electricities. So here you see a patient with Parkinson's disease who cannot control his hands properly. And after the doctor turned on an electrode which has been implanted in his brain, after several seconds, you can see that so the tremor disappeared. The patient can start talking, blinking, and most importantly, he can control his fingers very nicely. So perform this kind of delicate finger movements. So this is very amazing, because once we find the target in the brain which causes the problem, we can treat the disease by restoring the electrical properties in the brain. So this is a good example in Parkinson's disease. But what if we want to apply this technique to other brain disorders, such as psychiatric disorders? One critical challenge for this kind of application is that many psychiatric disorders are misdiagnosed and evaluated based purely on behavior. But so far, we don't have a very good understanding of how to quantify or evaluate behavior in an objective way. So here I want to propose a framework of translational neurophysiology, where we can look at the behavior and based on the behavior to identify the brain circuits, and then apply neurostimulation to treat the disorders. For this purpose, we need to use monkeys as our model animals, because First of all, monkeys can demonstrate very sophisticated cognitive functions, such as decision-making and other behaviors. And second, because the neuroanatomy of monkey is very similar to humans, and the brain size of monkey is very similar to humans as well. So when we want to apply any energy to stimulate the neural circuits, we can use the same equipment in monkeys and humans. So all these factors make monkeys an essential and very critical model animals for this framework. So, why do we need economic theories for decision making? So economics is a study of decision making, and it has been developed for decades. So economics provide us with a theoretical framework as to how to quantify and evaluate decision making. So the idea behind economic decision making is very simple. For example, when you choose between apples and oranges, let's say if you prefer apples over oranges, and economists assume that you assign a specific value to these options. For example, if you give a score of 5 to apples and score of 3 to oranges, because 5 is larger than 3, so that's why you choose apples. So it sounds very, very simple, but most importantly, in the past few decades, neuroscientists record from single neurons in the brain, and we do find neurons that encode these values in several brain structures. So here, this is an example of a single neuron that I recorded from my monkey. As you can see, there are 8 different liquids. So we know that these are plotted in the order of monkey's preference. So you see that after seeing a picture that predicts different rewards, these neurons kind of follow the monkey's preference very nicely. So it's quite amazing to find that indeed in the brain, we can actually identify neurons that's doing this kind of job, assigning the value to the options. However, to explain why different people have different preferences, or sometimes our preference might change tomorrow, it's a difficult question. Ultimately, we want to know how does or where does the reward value come from? It is a very difficult question, and in an economic theories, in the field of economics, it's like a mission impossible, because 2 Nobel Prize winners, Stigler and Baker, these are 2 economists. They wrote a paper in 1977 titled, it's a Latin, De gustibus non est disputanten. So in modern English, there's no accounting for taste. In this article, they argue that it is impossible to explain why different people have different preferences. So, but from the perspective of neural network, because they are single neurons that encode these values, these neurons must receive some input from other neurons. And these neurons need to take into account some critical reward components, not just choose randomly, right? So if we look at the animals in the field, especially monkeys, they usually look at their food options very carefully and choose them very wisely. So from the point of, so as I said, reward values might be influenced by many properties from the food rewards. For example, flavors, odors, temperature, textures, you name it. So that's why it's so difficult for us to choose food in a restaurant, right? But if you think, try to approach these questions from the ecological perspective, because the animals' food choices influence how much nutrients they get, and how they choose from the food rewards influence how well they can survive the evolution. So my hypothesis is that in the brain's reward system, there might be a mechanism to control nutrient intake. In other words, the animals needs to detect the presence of specific nutrient and choose these foods based on their nutrient requirements. So it sounds a bit straightforward, but it's quite difficult for the monkeys because in the monkey's world, there's no nutrient labels that tells the monkey how much nutrients inside the food options. So the monkeys need to derive this information from various sensory input. For example, in the oral cavity, the monkey can sense the taste and the mouthfeel of different food rewards. But how can they really tell the nutrient composition in the food and make decisions based on this information is still unknown. So in the following part of my talk, I will share with you some of my recent results demonstrating that monkey can actually choose from the nutrient components based on their own preference. And these preferences can be explained by the sensory, especially the mouthfeel of these nutrients. So first of all, let me show you a typical experimental setting in monkey neurophysiology. So usually our monkeys will sit in the primate chair like this, and they will hold a touch key or keyboard or anything that they can use to interact with the computer. So there's a screen in front of them. So to motivate the monkey to perform specific choices, we often give them juice or water, or fluid, some reward to try to motivate them. And during the decision making, we can place some recording electrodes on here, so to try to record the brain signals during this process. So the way we can measure subjective reward values in animals is like this. So in this task, the monkey needs to make a choice between two options. So each options is so monkey sees two different kind of pictures, and these pictures means different reward types. For example, apple juice or orange juice. The monkeys have been trained on these tasks for a long time, so they understand the meaning of the pictures. And from trial to trial, we use the bar here to indicate the amount of reward they can receive if they choose these options. So if we randomize the reward amounts and we ask the monkey to choose repeatedly between these two options, we can see how the monkeys make trade-off between the reward juice, the juice types and the juice amount. For example, usually the monkeys prefers high amount of rewards, but for juice types that he really likes, he might accept a small amount of his preferred juice types. So by looking at this trade-off, we can measure the subjective values in monkeys, which I will explain a bit more detail later. But most importantly, if we want to explain the reward values in terms of their nutrient compositions, we need to control the ingredients of the liquid rewards. So I kind of designed these 2x2 combinations of liquid rewards. So in other words, we have 2 sugar levels, low sugar, high sugar, and 2 fat level, low fat and high fat. So the monkeys make choices between these 4 rewards. So as you can see, overall, the monkeys prefer high nutrient rewards over low nutrient rewards. So that's a good start point, because we kind of show that so fat and sugar are really of positive value to the monkeys. Okay, so this is a bit the difficult part, so you might need to pay attention to it. So this is the tool we use to measure exactly what's the value of each reward in the monkeys. So on the x-axis, if we plot the ratio of the 2 options, For example, the amount of juice B versus amount of juice A. And the y-axis, we plot the choice probability of juice B. So when the ratio is very small, so for example, if it's close to 0, so the monkey won't choose juice B because he wants more reward, right? However, with the increase of the ratio, the monkey's choice probability will gradually increase at all the way up to 100%. So by looking at the specific point, when the monkeys are kind of, the two options are of equal value to the monkeys, this point is called indifference point. And this is a very important concept in economics, because this is the point when we know that the monkey value the two options equally. So for example, if the ratio is 3, this simply tells us that the monkey, 1 unit of juice A equals 3 units of juice B. So this helps us to quantify the reward values in monkeys without asking them how much they like it. Okay, so if we apply this tool to our nutrient-controlled stimuli, you can see that we can measure the value of fat and sugar in individual monkeys. So for example, here, this monkey value fat, the value of fat is like 1.5 times more than the low nutrient ones. However, the sugar values is like around 3. And most importantly, because we control the ingredients, we can know that how much the monkey, what's the subjective weighting of the each ingredients the monkey put in these ingredients. For example, these two monkeys, they have different subjective weighting to reward amounts, fat and sugars. So these help us identify the source of reward values in each monkeys. So secondly, I use a measuring technique from rheology and tribology to characterize, to try to evaluate how much this texture can explain the nutrient preferences we observe in monkeys' choices. So importantly, here, because we want to mimic the mouth field in oral cavity, I use a pig tongue to measure the friction of these rewards. So as you can see, we kind of validate the result in the literature. So with the increase of fat content, the sliding friction will increase. So which means that sliding friction is a good indicator of the fat content in the oral cavity. So if we include the oral texture in our regression models, we found that the monkey's fat preference can be completely explained by the oral texture. So it is a very good example because it kind of supports that the monkeys are able to derive the information of fat content from these sensory inputs. So most importantly, we want to know whether there are any neurons that are sensitive to these nutrient compositions. So here I will only show you some of the examples in the interest of time. So here, remember, we have 4 different stimuli which have either low or fat and sugar levels. So for example, in the first neurons, if we show the monkey a picture that predicts different reward types, these neurons respond to the high fat content, but not to the low fat content. So we can call it the fat sensitive neuron. Some other neurons respond only to the sugar levels, but not the fat levels. Most importantly, in the primate amygdala, we found some neurons that seems to process different information, these informations, by showing a slightly activation to fat, but a very, very high activation to fat and sugar together. So these neurons can sense the nutrient information and process this information. So with these neurons, it's like playing Lego in a neural network. We can build a neural network based on these all different neuron types. And these neuron types can detect the sensory input, evaluate the nutrient compositions, and then guide choices. So we even show that if we change the reward probability, so the monkey's choice will follow the choice probability. And if we construct a neural network based on these neurons, we can simulate or explain most of the animal's choices. So all these results create a completely new field called nutrient neurophysiology, which try to explain how in the brain animals can assign value to a specific nutrient and combine these nutrient values into a reward values to make choices. This is very important because these processes explain how animals adapt their food choices depending on their nutrient requirement. So, and after we know which part of the brain are responsible for these nutrient processing, we can try to change their activity by using different sorts of neuromodulation. So as I said, we can use a traditional deep brain stimulation by surgically place an electrode in the brain, but this is an invasive procedure, so we cannot do it in every human subject. However, in recent papers, we've seen that there are more and more non-invasive neuromodulations techniques coming up. For example, we can use electromagnetic fields or even ultrasound to change the neural activities non-invasively. So all these efforts create a very exciting application of these neurotechnology. And because in the brain, many decision-making involves several brain structures collectively known as reward system. And many of these psychiatric disorders such as ADHD, schizophrenia, depression, and so on, they have been implicated in these reward systems. So it's very exciting to link these behaviors and symptoms to a specific brain circuits in these brain structures, and then apply our modern technology to change the neuronal activities in these brain areas. So that's basically what have been done in the past few years, and I'm currently doing to bring my research into clinical applications. So I would like to thank most of my funders, my colleagues for their help. And if you have any questions, please feel free to contact me, email me, or just drop your questions in the chat. I think we have a chat room for you to ask questions. Thank you very much.
Thank you, Dr. Huang. And we have a few questions in the chat box. So the first question is, what brain regions are those value coding neurons recorded from?
Okay, yeah. So there are many neurons. I mean, value neurons have been identified in multiple brain regions. I think I can show you. So, so the neuron I record from is from the amygdala, which is a central area, part of the reward system. And these kind of value neurons has also been identified in prefrontal cortex and cingulate cortex and many different other brain areas. So that's why we believe that the neurons in these brain structures together form a specific neural network to control decision making. Thank you.
And the second question is why the reward value is sigmoidal curve instead of the straight line. And could you explain again, how do you measure it?
So yeah, so I think you are saying, talking about this. So here the sigmoid curve is not, so the y-axis is a choice probability. So that's why it's a sigmoid curve, because probability can only be between 0 and 1. But for reward values, it's a very interesting question, because imagine if a neuron encodes the reward values because of the limitation of the neuron, it cannot fire at 100 or 10,000 hertz. So the firing rate of neurons are also limited. So that's why we often see in these variant neurons, they kind of normalize the reward values to the range of choice options. So in other words, it is not the absolute reward value that matters, but the relative values, because you only need to know the relative values you can for decision making. Okay.
So the last questions I have in the chat box is in the middle amygdala. I'm not sure if I pronounce this right. So how do you decide where to record from? Is it the region connected to the taste inputs? Because,
okay, that's a great question. So usually when I do the recording, I try to record every to include most of the neurons in the whole amygdala. But because amygdala within the amygdala, there are different subregions of the amygdala. So after I record these neurons, because we can 3D reconstruct the location of these neurons and compare, well, whether they have different activity, what kind of information do they encode. So because I don't have enough time, but in summary, we do find that within the amygdala, different parts of the amygdala, they process different information, but they are all related to decision making. Okay.
Yeah, we just got 2 more questions.
Okay.
So, is there any reason like why the value neurons are preferred, and to like exist as positive to the encoding value distributed? And let me see. And the audience asks, like, you assume like for example, motor circuits will be highly associated with the value if a behavior is typical for certain judgments.
I'm not sure I understand your question very well, but it sounds like a complicated question. Or can we do it later? Is that okay?
Yeah. Or
I can look at the chat box and think about it.
Okay. Right. And another question is, does the firing rate increase after the monkey tastes the juice, or when they see the images or trying to make the decisions?
Okay, yeah. Initially, the monkey will respond to when they receive the reward. But after training, because we always show a picture and then give the monkey rewards. So after training, the monkey know which picture predict the rewards, and then the response will move from the juice delivery to the purely visual presentation. This is very important because imagine when you do shopping in the supermarket, you need to know the value of the options once you see the option, right? Not just after you eat it. So the monkeys are able to do that, and the neurons kind of can transfer the value signals from the actual juice delivery to the visual stimulus. Okay.
So our last questions. So could some monkeys prefer too many nutrients compounds? So these compounds are actually like detrimental to the monkey's survivals.
Yeah, that's very important. So that's actually what I'm looking right now, because I want to understand what's the mechanism. So for example, in our monkeys, we show that monkeys prefer fat and sugar, but do these preferences change with their physiological status? For example, if we feed the monkey too much sugar, can the monkey stop eating more sugar itself, or there's just no such regulation in animals? So far we still don't know, but that's I'm looking at it right now. So I don't have the answer right now, but that's a very important future direction of my research. Thank you.
Thank you, Dr. Huang. I think that's all the questions. And if any audience have more questions, please feel free to send an email or use chat box to ask more questions to Dr. Huang. And thanks, Dr. Huang's exciting research to explain how the neuron or brain to make the decisions. And the next talk, we're talking about a different topic, not totally different, because the reason how the neurons affect our behavior will be we need to dig into the cells, and we can see there's a cytoplasm and the nucleus, and inside the nucleus there's one thing called a DNA, and DNA will be the key components to determine what we behavior when we give birth or when we are born, to decide how tall we are, what we are looking. And more probably a lot of people have known this a couple of weeks ago, there's a paper published in the Science, finally human complete the sequencing for the human genomic DNAs. And why the genomic DNA is very important, because in the futures the precision medicine will be the trend for the future biomedical the research or the therapeutics, and the genomic DNA sequencing will be the key elements to help the doctors or researchers to develop a new therapeutics to treat any genetic disease. And I want to introduce our next speakers, 𝗦𝗽𝗲𝗏 Chen, and he studied computational genomics at the Department of the Computer Science at Johns Hopkins Universities. And he designs the DNA sequencing processing methods that reduce the reference bias. And his primary work including the sequence alignments, leftovers, the variance callings. And I'm pretty excited to see his research, because it's the previous the publications in the science will be the huge milestone for the bioinformatics. So let's welcome 𝗦𝗽𝗏 for his talk.
Hello, can you hear me clearly?
Yes.
Thank you, Jason, for introduction. So I'm 𝗦𝗽𝗏 Chen. Today I'm going to talk about my research and a little bit more introductory for the genomic data processing methods, and I'll talk about how do we improve genomic data processing by reducing reference bias. And so in the next 20 minutes, I'm going to introduce what is genomic data processing, what's the main concepts of it, why is it important. And then I'm going to talk about, as Jason mentioned, the first complete human genome. And this is a very exciting advancement in our field. And I'm going to talk about my method to leverage the improved quality of the first complete genome while remain compatible with the existing data processing methods. And the last part, I'll briefly talk about the future directions of genomic data processing, which is we want to include more population information in these processing methods, because the current method is a little bit limited to with lack of diversity, and that will become problematic in the future, or it is actually a problem right now. So these are the main agenda that I'm going to talk to you today. So first, let's talk about what is genome and what is genomics. So these definitions are found from the National Human Genome Research Institute, that the genome is the entire set of genetic instructions found in a cell, and genomics is the study of the genome. So genomics is actually they're inherited DNAs, germline mutations, something like that, and it also includes environmental factors. But I'm going to not talk about environmental factors today, but I just want to kind of note that like genomics is not only inherited, it is a combination of like inherited information and also like environmental factors. So genomics is a very broad field. So we have a lot to study like in genomics. And today I'm going to be a little bit like narrowing down to a specific like subtopic, which is the processing and the analyses of genetic differences. So the problem is something like how genetically different is an individual compared to others. For example, we want to know like from textbooks in like middle school or high school biology, we know that if we have a additional copy of chromosome 21, that is like chromosome 21 trisomy, we'll get like Down syndrome. And we have also like a lot of like large scale like genomic disorders. So that's like important, and that's like very bad for the patients. And there are also like smaller scale events, for example, a point mutation or like a relatively small mutation in a genome can affect like the kind of the behavior, the phenotypes of an individual like very dramatically if like such mutations are in critical regions. And we know like SARS-CoV-2, like we talk about different variants like every day, like a like virus strain can mutate and can have like different mutations, and they have like those like evolutionary like final genetic structure. So those are like all very important questions that we want to ask. And now like because of COVID, like I think people have like plenty of attention like in the genomics field, because like we know that variants can be very, very important. So genetic differences is something that we want to study. And the next question is, okay, we want to study it, but how? And this is like hard actually. Like, so if like we have a kind of black box machine that can sequence the entire genome of the target like in one run and with like high quality, that would be fantastic. But unfortunately, it is not available at this point in 2022. So sequencing is like we can do very high accuracy sequencing, but we cannot do it in one run. So so far, at least for human, like human is kind of a relatively large genome. So for a human, it is impossible to kind of sequence a human genome in one run with very high accuracy. So we have to do a lot of pre-processing in order to make the kind of genetic difference finding possible. So here I'm going to introduce like what is like a genomic data set looks like. So if you have done genomics, you probably like have seen something like the screenshot I showed on the left. This is called a FASTQ format. The format name is not super important, but like what we are having are like small pieces of DNA. For example, the most like common sequencing machines like have like a lot of reads. A read is a like small DNA segment, and it is usually about 150 base pairs. So 150 like characters, ACTGs, like the total length is 150. So each 150 segment is called a read. And so like recall that like our end goal is to understand the entire, say, human genome and want to spot the differences so that we can compare. And it is like really hard for us to get like from 150 to the entire human genome. The size of a human genome is like 3 billion. So it is 3 followed by 9 like zeros. So that's like a big number, and like it's a lot bigger than 150. So the very first thing we need to do in like genomic data processing is we need to solve this like genomic jigsaw puzzle. So it's like my cartoon figure here. We got a lot of small pieces and we want to assemble them together, so then we can find the differences. So this is like the very challenging part like of genomic data processing. So to do it, it takes a lot of like time and efforts and money in the beginning. And this is like the Human Genome Project, which is initiated by the Department of Energy in the United States. And there was a like huge international collaboration that was happening around like 90s and 2000s. And so like that's a big international effort. And the Human Genome Project is like in most people's biology textbooks, and it was in mine too. So it was completed in 2003. So scientists claim that, okay, we have the human genome assembled. Like, so we got a big picture of like what a human genome looks like. And this is like a big milestone of like biology research history. And we can now study and compare like human genome versus other species, or we can look at like a lot of elements. We can look at like how like the human genome is composed and what are there like inside, and we can dig deeper into it. And additionally, having one human genome also like makes it a lot easier to kind of help us get more, because we have the assumption that humans share very high, is more than 99% genetic diversity. So when doing our jigsaw like the jigsaw puzzle problem, having one human genome makes things a lot easier, because we can ask the computers to compare. We still have like for like other donors, for example, we have a patient coming and we want to sequence them, and we still have those like 150 base pair segments, so they are still small segments. But what we can do is we have another strategy of solving the jigsaw puzzle problem. In the beginning, we have to assemble them from scratch. We have to look at the shape, look at like the similarity between each pieces. So that is really, really time consuming. But if we have like one kind of like assembled human genome, we can use it as a reference. And it's like we solve a jigsaw puzzle, we look at the case, we look at the image, and then we have like small pieces, and we kind of try to match like the image like on the case, and that will be a lot more efficient than the previous approaches. And this is called alignment. So here is a like common like genomic data processing pipeline. So it starts from aligning like the DNA reads or small segments to a reference genome. So it's like this. We have like small segments here. We see a like rabbit face. It's a cat. So we see a face of a cat, and then we try to like search through this like image, which is assembled like by the Human Genome Project, and we find, okay, like the cat face is here, so we can like put this piece here, and then we know like some information about it. And this is the first step of genomic data processing. And the second step is we want to know where the differences are. And there are other applications too, but today we're going to focus on like genotyping, which is to find the mutations like of this individual versus others. So this is the spot the difference game. So like it's like playing a lot of games in the processing like pipelines. So we want to know like, okay, like we have this like this image from the sample, and we have this image from the reference, and where are the differences? For example, we know that there are like different numbers of bananas, like we have a trophy here, but a like kind of like something different here. So, and the time is different. So these are like mutations. So imagine it is like genomes. So we have like mutations in different genes or in different like regions, and then we can spot those differences, and we can perform downstream analyses like given those differences. So everything sounds good, and like the genomics has like huge improvements and like a lot of more biological studies, and we are kind of using genomics and like basic science in like medical research, in clinical, in like gene editing, gene testing, something like that. So this is like very, very influential, but we still have problems. Like it is not perfectly solved. Like genomics is still far from a like kind of solved problem. So one of the major problem is that the human reference was not complete. And we mentioned that like people spend lots of like efforts in the Human Genome Project to kind of compile a very good high quality reference genome. But like the latest release was in 2013, which is called GRCH38. So GRC is like the Genome Research Consortium, and H is human. So the like 38 version of human is like published in 2013. And there are still like 875 gaps For example, those like purple areas like are the gaps in the reference. So these are like about like 200 million bases that are missing or unlocalized. So going back to our jigsaw puzzle analogy, like we have some black regions in the photo that we don't know like what they are, or they're like badly quality, something like that. So the information like for the reference genome in some these like purple like regions are not discoverable. So this is like not ideal because those are important regions and we want to study them. And like there are other like small or large scale errors that we don't know, like the reference quality is not great. So we don't know how to deal with them. So this is a major problem that we want to solve. And fortunately, like the technology has improved dramatically over the past 2 decades. And one important advancement is called like long reads. So like, as I mentioned, like the sequence data, the data that we can sequence is usually 150 base pairs. And like, but the latest technology, we can sequence about like 20,000 base pairs and with like really, really high accuracy. So again, that's back to our jigsaw puzzle. Like we have like, initially it was like a several million piece puzzle, which is super difficult. But like if we got like, if we can reduce the number of pieces by like several orders, then things are a lot easier than what we were facing in the past. So the long reads with like high quality significantly improved the quality of human assemblies. And that makes this possible, which is like published earlier this month on April 1st, which is an interesting date, but like this is the first complete human genome that is like ever sequenced. So there are lots of like hard works here. Like this paper had 100 authors. So this is a kind of really, really like incredible job by the scientists. And like, I'm very fortunate to be a member of it, but like this is like very big international efforts. And you can see on the table that is like published in the paper that there are no unplaced bases, there are no gaps here. And like we kind of increase number of genes predicted by about 5 to 5.7%. So we have a lot more in the new genome than the human genome project reference. So we have more to study because of the advancements like in technologies and like the efforts by the scientists in this group. So this is a very exciting milestone in our group. And in the field, then we want to ask the next question is like, okay, we got this exciting new genome, then how do we fully utilize this new genome? But unfortunately, this is really difficult because like in the past, the transitioning between references has shown to be a really, really hard task. So as I mentioned, before the complete genome, the latest one was GRCH38. It was published 9 years ago, but many analyses now still use GRCH37, which is an earlier version of the human reference genome. And that is not ideal because genomics is a very fast moving field, and 9 years in genomics is like very, very long. We like very few methods like a decade ago was still like applicable now to the data analysis and something like that. So that's a long time, and that's not like what we want to see in the community, that we have an improved reference, but people are not going to use it. So which means like we are generating a lot of like false results using the old reference because they are like relatively lower quality, and that is not great for like biological discovery and for like clinical uses as well. But like, why don't people like to switch to a new reference? And it's primarily because of the huge costs to rerun all the analyses. As I showed in this like screenshot, people are complaining that, do we really need to rerun all the analyses again? And like maybe in the next 5 years, we'll have a new reference, and do we need to do it again and again and again? Like people really don't want to do it. And that's like, there's also a lot like difficulties in like validation and interpretations of the results. So changing a reference is something like that is there are benefits, but it is also super painful. And also one thing I want to mention is we have a lot of existing annotations that is purely based on existing reference genomes. So these annotations include like gene annotations, where are the exons, where are the introns, or like kind of disease association annotation, something like that. So there are lots of, as I mentioned in the like several first slide, that genomics is a very comprehensive field, and we use we annotate the different regions in a reference genome to tell like other users that, okay, this is a functional region that is like associated with some other phenotypes. And these annotations are usually reference specific. So if we want to switch to a new reference, we lost all these existing annotations, and that is not great. So I'm going to talk briefly about my method, which is called 𝗧𝗶𝗺𝗲2. What it does is to lift reads from the new reference to an old reference. And in the genomic data processing pipeline, it is here in the alignment step. So the goal is we want to adjust genomic coordinate systems, because as I mentioned, different reference genomes have different coordinate systems, and that means like the annotations are not compatible. So our strategy is we align reads initially to the complete human reference, because it is the best reference available at this point. So we align all the reads to this like new reference. And then like my methods kind of doing a leftover process to migrate all the alignments based on the new reference to an old reference that have like rich annotation support. So this is like what my software tries to do. And one kind of important concept is we have like about, as I mentioned, like previously, we got 200 million base pairs that were like unplaced or unlocalized in the old reference. And we still have data sequence there. Like those are like real genomic regions. We just don't have the reference for them, but we will sequence reads from those regions. And in the past, because we don't have those like sequences in the reference genome, so we'll just like put it to random places, and that will result in false positives and like kind of waste of computational resources. So that is not ideal. And so what I'm doing is because we use a dual reference system, so the reads in those like new regions are just staying like on the new genome, and we didn't migrate them to the old genome. So that is like reducing the number of reads that we want to process and reduce the number of false positive mappings. And in the end, like the workflow works very well. It reduces 10 to 20% variant calling errors, and it supports both short and long reads. So we're trying to support it for like the common genomic data processing pipelines, and like 10 to 20% error reduction is dramatic. It's something like really, really substantial. And for example, we can look at this image, like it's hard to zoom in here on like Keynote, but the idea is like we got two tracks. The top, this like kind of colorful track is like the alignments using an existing reference, and the bottom one is the alignment using our method. And gray means like things are kind of identical to the reference genome. So usually gray means pretty good. And there will be a little bit like mutations, which is colored in non-gray colors like red, blue, something like that. So if you see a colorful bar, it means a mutation. And if you see this track, we have like a moderate number of mutations, which is right. But like if you see this track, we have a just crazy number of mutations, which is not right in the reference. So this image shows that like my method reduces the number of errors dramatically in this specific region. And that is because of the benefit of the new reference and like my kind of selective approach to kind of try to map the reads that is like needed to be mapped. So that is the first part we talked about to leverage the improved quality of the complete human genome and to make it for better genomic data processing. And the next step, I will briefly talk about the next challenge, which is the lack of genetic diversity in the system. And we know that like we got like one human genome as a reference, but that's like one data. But actually, like for each like healthy like human individuals, we're deployed. So we got 2 genomes. Like we actually got a pair of like genomes for us. So it is like kind of impossible for one reference genome to represent an individual. And in like a population, we got like millions or hundreds of millions of genomic variants in the population. And it is impossible for one human genome to represent all of them. And that like introduced challenges because, for example, in this region, we know if we got a kind of teeny jigsaw puzzle here, a piece here, there's no matching this like ball or trophy on the reference genome. And if we see something blue, we might try to map it to the window region, and that is apparently a mistake. So the lack of diversity in the human reference is making problem. That is not only for human, but for other species as well. And so first, for my method, I developed a method called reference flow that is also in the alignment step to include genetic diversity in the data processing system. It was published in Genome Biology 2021. And the idea is we got like reads in different groups. So some reads that can be, like most of the reads can be successfully mapped to the reference genome, but some of them need kind of extra care. So we need like extra efforts for them. And we use like a set of designed population references to handle those like difficult reads. And we kind of realign them like in a selective way, a little bit similar to the Leviosan workflow. And it results in like high accuracy and much more efficient than the best competing method. For example, my method is here, and this is like the optimal result, and this is the best competing method, and this is the baseline. So we show substantial improvements like over other methods, also remain computationally efficient. And also like including genetic diversity in variant calling is also helpful. So this was like during my internship in 2020 at Google. So we developed a variant calling method that includes genetic diversity in variant calling, and it reduces about 5% of the errors compared to the second best method. So we showed that like for both alignment and variant calling, having the inclusion of diversity can be beneficial. And this is the last slide. So today I talk about like genomic analyses, and they are confounded by reference biases, including assembly artifact and lack of genetic diversity. And like I mentioned that like methods like that is kind of designed for such like artifacts can be addressed and improve genomic data processing accuracy. And I thank my kind of collaborators, advisor, co-authors, many people here, and thank you for your attention.
Thanks, Natrins, that great presentations. And we have one question from our audience. The question is, since the reference is critical in the whole human genome project, and the audience wondering why this specific reference was selected in the beginning, and what's the requirement for the reference, and if there's any like the more like representing.
So for the human genome project, they kind of collect volunteers that like, so I think about 70% of the human genome project reference is from one individual that has like, that was a African American male. So a lot of data was from that specific individual. So it is hard because like picking a reference, what it is actually doing is to find a person to represent human, and no one like is that good to represent human. So it is a little bit arbitrary, and there's a historical process. And in the complete human genome study, like the data was actually not from a healthy human, it was a little bit from a kind of tumor cell, but like that tumor cell has like very kind of interesting characteristics. It is like haploid, it has like only one like the paternal and maternal copy, because like there was issues during like that was a kind of tumor, so the mother DNA was not there like in the egg, so something went wrong with the egg, so it has like duplicated DNAs from the father, so that makes things kind of easier to to process because we are kind of reducing complexity, but that's not from a healthy individual as well. So I think you raise a very good question, and like it's still pretty challenging like at this point, and I think it will still be challenging in the future.
Another question from the audience is the number of genes increased by 5.7% in the new reference genomics. So what type of genes are like significant increase in the new genomics?
Yeah, I think that's a very good question. And I don't have a perfect answer to it, but like they are mostly non-coding because like the newly introduced regions are mostly like centromeres and telomeres. So those are like not protein coding regions, or they're kind of more in the regulatory elements. So I think we have limited understanding for those regions and have limited understanding for those like predicted genes. The number of genes was predicted, but not like from functional assays. So we have a lot to look at like for these data.
Oh, we got another question. So can you explain why the old reference, the 38, has more like annotation than the latest one?
Yeah, because it was there for a long time. So like we have the reference and people like analyze the reference and have like add annotations on it. So it was there for like longer time than like 1 month. So we got like more annotations for the old reference. And yeah, that's the answer. That's not like, that's kind of a practical concern, but that's not a like kind of real science like reasons.
So the last questions, besides the gap is filled in the updated reference, is there any changes in the sequence of the known genes in the new reference?
I think it actually, there's plenty of them. And I think this is, we do have a lot of genes that were kind of changed a little bit, but like changing it for like a number of base pairs, like 1 or 2, actually doesn't matter that much. But there are some like kind of segmental, like large scale. For example, there are genes in like repetitive regions. So repetitive regions in genomics are really hard. And the new reference is kind of resolving some of the repetitiveness, like because in GRC38, there are plenty of like false duplications and that are resolved in the new reference genome, and that would affect data processing in some specific genes nearby. And so it's hard to say that like, does it change the gene kind of directly? But like if considering the entire reference, there are plenty of genes that like the data processing results would be different, like by using the new reference versus using an old reference.
I think the time is up. So that's all like today's like the seminars, and hope everyone like enjoy these 2 presentations. And if you have any questions, feel free to use the linking like to contact the speakers for more questions. And hope everyone enjoy the rest of the weekend. We will see you in our next event. Yeah.
Like Annie?
Oh, okay. So can everyone turn on your camera so that we can take a group photos?